Okay, I'm going to try this one last time. For some reason, it's not uploading. So I'm going to quickly show you how to convert from DNA to RNA and then find the amino acids in case you're having problems, okay? I've had a lot of text today. So the first thing is you need to have this sheet that I gave you, all right? It's going to help you find the codons. I'll show you how to use the round one. I prefer this one. Doesn't really matter. But as we're trying to find our codons, okay, we're going to look at first base, second base, and third base. So I'll show you that in just a second, okay? But I'm going to show you this one first because once I write on it, I can't move it. So this is the circle one that's on your paper, and it's in your notebook. So if you have a 3-letter codon, which is how the ribosomes are read, the ribosomes reads 3 nucleotides in a row, and that's called a codon, okay? So your first codon, so let's just say I have AUG. My first letter is going to be here in the center, so I would go to A. My second letter is going to be in this circle right here, so I would go to U. And then I'm going to find the G, okay? So you can see either U, C, or A is going to give me isoleucine, or G is going to give me methanone, right? Methanone is also the start. On mine, this one here, it tells you that it's start, okay? As well as that. So real quick, how do we do this? Here's number one. You just translate it or transcribe it just like you're doing DNA, okay? So I have a T. Just remember that there's no T on the messenger RNA. So wherever I would want to put a T, I got to put a U instead. So complementary base pairs, apples grow on trees, cars go in the garage. So T is going to go to A. Now I have an A. Normally I'd put a T, but because this is going to be messenger RNA, I put a U. C goes to G, C goes to G, G goes to C, A goes to U, T goes to A, G goes to C goes to G, A goes to U, and then I got a C, A, U, T goes to A, C goes to G, A goes to U, A, and G. Okay, so those are my nucleotides. Then I'm going to break these up into 3 letters each. So I've got AUG, draw a line. GCC, draw a line. And then just draw a line between each one of your 3 at a time, okay? Now I got to go back and I got to read it. So if I have this one right here, okay, see if I can do this. It works better on my iPad because you could just see better, but not much I can do about this. Okay, so here it is. I don't know if you can see this, but just kind of follow along with me. My first one is A, and then the second letter is U, so it's going to be right here. Oh, sorry, can't read. A, U, and then so I'm going to come down here, and then I got G. So it says start methanonin, okay, methanone. So MET. So up here, I'm going to put MET, which is also my start. Now, sometimes you might have letters in front of this, okay? It doesn't really matter. It's going to start reading. Where you care about is where you see this AUG, okay? To my knowledge, this is the only one that codes for start, so that's what you're going to look at. There's a couple that code for stop. And then I've got GCC. So I'm going to come here, and I'm going to find, what did I say, G? So here's my G. I come across to C. All my GCs are going to code for alanine, which is ALA. So I'm just going to put ALA. My next one is UAC. So here I am. I'm going to go find U. I'm going to find A. I'm going to find C, and that's going to code for tyrosine, TYR. Okay, so you're going to keep doing this. GGU, GU, those are all going to code for glycosine, GLY. And then I got CUA, so I'm going to find C, U, and A, and all my CUs are going to code for leucine, which is LEU. And then I got GUU, so I come over here, I got G, I find U. All my GUs, no matter what the letter is going to code for valine, so that's VAL. And that's good because if I have a minor mistake, I won't have a I won't have any issues with my amino acid because all of these nucleotides or all of these codons code for the same amino acid. Okay. And then my last one is UAG. So I go U, come over here to A. I have G, which means stop. Okay. And then I'm done. So let's say for argument's sake, I had letters before on my DNA. I had letters before that T, and I had letters after the last C. I would go ahead and do my complementary base pair for the messenger RNA. But when I'm finding my amino acid, these are what I'm going to look for. I'm going to do those two and everything in between. Okay. Now, there's a question on page 19 or 20. I think it's page 20. And it asks about anticodons. Okay. Well, the anticodon is just going to be the opposite of the codon. Okay. So these are called codons. When I do my messenger RNA, it's called a triplet codon. Okay. So let's just say I'm going to, instead of amino acid, I'm going to write anticodon. All right. So if I did my messenger RNA, I would have A, U, G, then I would have G, A, C. That's all I need to do. Okay. So if I'm doing my anticodon, what goes to A? U. What goes to U? A. What goes to G? C. What goes to G? C. What goes to A? U. What goes to C? G. That's the anticodon because then it's going to go and find this amino acid. Okay, so it's got to be the opposite because it's got to attach to it and bring it back, right? So we can talk more about that tomorrow, but just to kind of give you an idea. If you have any questions, I'll be up here a little bit, but I can always respond to your text. Okay, good luck.